my name is Andrew Angel. I'd like to thank the board for coming to review my proposal for continued funding for the genetics department. I am a spokesperson of the genetics department, and I'd like to give you a rundown of what we're doing over there and kind of a description as to why it needs continued funding. So we're working on CRISPR. That's a genetic editing tool, and it's also an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. But in plain English, it's essentially a tool that we've developed that allows us to cut and paste genes. We initially discovered this early 2013 when we looked at E. coli, and we found that the bacteria had developed this as well. They had an innate immune system that allowed them to cut specific sequences of DNA. Now, I know many of us have been affected by some sort of genetic disease, and it's difficult because a lot of the times there's not much you can do other than treat the causes of what's happening. But CRISPR offers a solution to that. CRISPR, essentially, what it does is it's inserted into the body, into the cells directly, and it can both identify and remove, as well as splice together, faulty genome sequences. Essentially, what this means is that we put it into the body, a newly developed infant, and we tell it to target specific genes that we've identified as bad, whether that be a faulty cancer gene or a gene for another genetic disease, but we identify it as bad and harmful to the body. Next, the CRISPR technology goes in and it splices that specific sequence out and it mends it together. So it cuts it out and it fuses the DNA back together after it cuts out. And essentially what this allows us to do is prevent any sort of genetic disease from ever manifesting. So if it's identified that they're going to have cancer later in life and it's unavoidable, this is now something that we can treat with CRISPR, and it's now something that we can essentially put behind us if we get to that point. Now, early trials look great. We've had great success with it, but we still do need funding. There's still a lot we don't understand about CRISPR, but within the next 5 years, we could be at the point where we could essentially copy and paste genes into the human genome, something that's never been heard of, and it's never been heard of at this efficiency. So CRISPR is not a technology. It is a specific protein that has been bent a certain way to interact with the human genome. So essentially, we found it in bacteria, and we tweaked it slightly, and now it responds to how we want to respond it to. Now, I urge you to continue funding us, because without continued funding, this technology essentially becomes obsolete. It's only applicable at the university level. I'm unsure of anyone else doing it other than major universities, and I would love for UNM to be pioneering on the forefront of the next genetic treatment. This, quite frankly, could definitely save lives. There's no doubt in my mind that it could save lives. Thank you for your time.